Thank you, Dr. Newby. Okay, up next, we have Dr. Dennis Koh from Duke University. And he's going to have a very interesting talk that has kind of a different take on exploring genetic risk and MS.
Okay, great. Thanks, Sabrina. All right, so it's a pleasure to be here today. I'm going to tell you a little bit of the story of how my lab, which studies genetics of infectious disease, has somehow come to study MS a little bit in collaboration with Simon's lab. And everything in my lab starts with diversity, right? So you look around this room, no two of us look alike. We're different in lots of different ways. We're different in how tall we are, our intellect, our athletic ability. And one way we're different, which we can't really see, is how different we are in our susceptibility to infection. Right, so this is a picture of a bubonic plague, right? And this person's doing quite bad, as you can see, but the people around him seem to be okay. So what causes those sorts of differences? So one way that people can now ask that question from a genetic standpoint is we've heard a little bit about using these chips to look at our genetic differences. And this is through what's called the genome-wide association study. Right, where we have two groups of people. You have people who are patients and non-patients. You get their DNA, just as we're collecting the MURDOCK study, and then we look for the differences in the frequency of these two groups in terms of their DNA, in terms of single nucleotide polymorphisms, or SNPs. So, you know, as everyone in this room is aware, this has been done for multiple sclerosis, and Simon has been involved in many of these studies, where you have a group of MS patients and controls, and you try to figure out where in the DNA are there differences that affect risk of multiple sclerosis, okay? So this is a map of that risk. So you can think of the same thing with infectious disease, in that there are genetic differences among us that lead to differences in how susceptible we are to different things. So for example, many of you probably learned in high school that if you have sickle cell trait, you're also protected against malaria. And similarly, there's a common mutation that probably about 10% of the people in this room have in the CCR5 gene. So this is a gene that we all have. Some of us have a little deletion, a little bit of DNA missing there, and that causes protection against HIV infection. Right? So there are these common genetic differences that protect against infection. And this is important because, you know, everyone here is thinking about, can we actually get to cure somehow? And so, for example, inhibitors of the CCR5 protein are actually used to treat HIV, right? So this is a discovery that starts with basic biology and understanding our genetic differences that has actually led to a drug. Okay, so these GWAS studies, these genome-wide studies looking at our DNA, are really great to connect these genetic differences to human health. Right, so we can connect genetic differences to HIV risk or to multiple sclerosis risk. But one of the strengths of the MURDOCK study, right, is that we try to fill in these boxes in between. It's not magic that this connects to this. It's the fact that genetic differences affect genes and proteins and metabolites, like Simon was talking about. And this affects how our cells function, and that affects human health. So the approach my lab takes then is a little bit different in terms of genome-wide association. Instead of trying to connect the genetic differences directly to human health using patient volunteers, we take cells from different people and try to ask the same kind of question. So we try to do cell biology to connect genetic differences to variation in cellular processes. So the way we do this is this approach that I developed called high-host. And basically, instead of lining up people and exposing them to pathogens, which most people would frown on, we just get cell lines from different people, put that in a dish, and then we add different pathogens to that. Right, so the idea then is that we can map the genetic differences. Sabrina actually used the same SNP figure before, right, to try to map these genetic differences onto variation in how our cells react to different infectious agents. Right, and the advantages of this, right, is we can do experiments on these cells, right? Once we find an association, we can then ask, Let's test that experimentally in the lab and see what happens. And then, of course, we want to then link that eventually to what does this actually mean clinically for people as opposed to cells. Okay, so the story here for my lab starts with salmonella. So many of you hear about salmonella in news. It can cause gastroenteritis. So there's like outbreaks every year, tainted peanut butter or lettuce or whatever else. So it can cause gastroenteritis. It can also cause a more serious infection called typhoid fever. And some of you may have heard of the most famous carrier of typhoid fever, which is Typhoid Mary, who was working as a cook in the early 20th century and got plenty of people sick because she wouldn't stop working as a cook. And as you can see, she's serving up death in a skillet over here. So what infections like salmonella have in common with things like MS is the fact that the immune system is involved, right? So the immune system helps recognize foreign invaders such as the salmonella coming into your body, but it can also have negative consequences as well in the form of autoimmune disease, right? So this cartoon here illustrates that, right? So you have your immune cells, they're like, intruder, destroy, and there's no salmonella around here, it's just another one of your own cells. So this person's like, what? But I'm one of you, right? So don't listen to her lies, right? So there's this sort of dual role of the immune system in terms of recognizing foreign invaders like salmonella, but also making sure that your own cells aren't being attacked, okay? And you can see that these immune cells are communicating here. And the way these immune cells communicate are through proteins that are called cytokines. So this is a complicated diagram which is just showing that there are different kinds of cells in your body. And like these cells here are talking, cells talk by producing these different cytokine molecules you see here, okay? So in my lab, we decided to ask, if we infect cells with salmonella, we just have cells from different people, we infect them with salmonella, what kinds of things are they saying to each other in terms of cytokines? Okay, so we measured all these molecules, and what we saw was that, this one seems to be going up the most, this interleukin-10. Okay, so what is interleukin-10 or IL-10? Going back to this analogy here, if you had a cell coming in here, IL-10 is an anti-inflammatory cytokine. Okay, so it's a protein that looks like that, and basically what it says is, let's all chill and be friends. No more inflammation here. We could probably all use some IL-10 after the election is done tomorrow, I'm pretty sure. So the idea then is, let's measure this IL-10. Let's measure how different it is among all of us and try to find the genetic differences that explain whether people have high levels or low levels, okay? Right, so we tried to measure this in hundreds of cell lines from different people. And then we ran this genome-wide association analysis, and that gives us what I think of as, what it gives us is a map of where all the genetic differences are and how associated they are. How well can each of these different SNPs explain the amount of salmonella-induced IL-10 that's going on? Okay, so I still haven't come to connecting this to MS yet, but that's coming up now. So the idea is we can take this and use it as a signature for salmonella IL-10 production, salmonella-induced IL-10 production. And we have the same sort of data for MS, right? These are two different signatures of of different diseases or different phenotypes. So then the question we ask is, can we just compare those two signatures? Let's compare this signature with this signature and see how similar they are, okay? And the way we do that is just looking at the list of SNPs that are associated with each trait and asking, is the overlap between those two lists just random, what you would expect just by chance, or is it more than that? Okay, so we developed some software to do this. I don't think I'll dwell on that. And then we basically just found that there are 6 SNPs that are shared between these two things, okay? 6 genetic differences are associated with salmonella-induced IL-10 and are also associated with multiple sclerosis. Okay, and these are the SNPs here. There's 6 of them, And what's interesting is that in 5 of the 6 cases, the SNP that causes, that is associated with higher risk of multiple sclerosis is associated with lower levels of IL-10. Okay, so then this leads to a couple questions. And this is where we started thinking about it, MS some more. Right, the questions it leads to is, does the low IL-10 lead to a higher risk of multiple sclerosis? And this is something that people have looked at previously, but the data is pretty, it's pretty all over the place for that. So we thought, well, it'd be nice if we could look at that in people, as well as the mouse model that many of you, that we've heard about earlier today. So the MURDOCK study basically made it possible for us to ask that. Without that study, we probably would have just gone back to studying salmonella some more and left it at that, that this is kind of interesting, but we can't really test it. Now, sort of the crazier idea, and I'm someone who really likes crazy ideas, so I like this idea, is does getting food poisoning with salmonella and then having your immune cells producing all this IL-10, does that actually reduce your risk or severity of MS? So we don't know the answer to that, but it's just an interesting thing to think about based on what we found. Okay, so talked to Simon. He said, yeah, we can measure that. Let's measure IL-10 in